That's for sure.
Hi there. Mike. Get down in here. Fish room.
You got in early this morning. Ready to go.
Laura Jo was there. I was hoping I didn't have to do the alarm.
I set it up for him.
She was there, so I didn't have biggest concern.
So beautiful.
Where's yourself?
No raisin.
Oh, yeah.
You wake up beforehand.
4:30.
You all ready? You studied for this session?
Yeah. Test at the end of this.
Forgot to tell you how to study ahead of time. What was going on?
Just by the title, I know I'm going to flop.
I can give you the title. I got that.
Oh my god, I thought this was two of them. It's all one.
It's all on. This is the annual 3-hour session. Get the memo. There you go. Taking a break for lunch, but it should be on a veg diet. Patients and other. Yeah, it's kind of an interesting website. I haven't seen the actual book. He makes it.
Mauna Kea or whatever, Volvo, which one do you talk about?
Also,
Yeah, that's right. Way too old.
organic
It's dirt road.
vegetables, washed up,
Yep.
no preservatives.
You would do it.
Well, I don't know. Well,
Nothing would hold you back.
if it's a rental car, what the hell?
Who cares? What time we did?
go ahead and get started. I gave David a pretty difficult topic after I created this topic. Firstly, I got it wrong. One of the genes, I put GATA1 actually is the original title is involved. I looked it up later more in red cell development. I think it's got very little or nothing to do with T cell development. I was just mentioning to him, and I think he's going to make the point, it's amazing how this field, for those of us old guys in the room, that none of this was known when we went into this field, and in the last decade, just understanding the genetics of T cell development is absolutely amazing. So I'm sure he's done a great job. His third aim, if you take a look at it, is survive going through an hour of heavy-duty basic science at 7 in the morning. So I know he'll spice it up and make it enjoyable for us.
This basically has nothing to do with what I'm going to talk about, but I think it's a nice way to start it. When Mephisto asked first asked...
David, speak
yes.
up as loudly as you can. Nick's as deaf as I am.
Bring the microphone closer to.
So basically that's a quote from Goethe about Faust. When Mephisto asked Faust to sign a contract, he asked him to sign it with his own blood. And the reason was that because blood is a very special juice. And I guess we can agree with that. I'm going to talk about master regulators of CD4 T cells differentiation. I left FOXP3 out of the picture today because I think he deserved a different talk maybe next year. I'm going to talk about T-bet, GATA3, and or gamma T. Or if we can continue with the GATA team, the GATA team, these are the souls of a young CD4 T cells. Still no disclosures, probably will never have. And we'll start with some definitions. So, signalophobia. Signalophobia is an extreme or irrational fear of intracellular signaling pathways. And you can also use it in a sentence like David suffers general signalophobia, with more severe symptoms when it gets to stat phosphorylation and something like that. I'll get back to that later on. So don't
That's
worry.
probably a real diagram.
It is.
Unfortunately.
The second is whenever you hear me say, as you all know, it means that that was the first time I actually heard about it. This is a real one, transcription factors. So what is a transcription factor? All these T-bet, GATA3, and all gamma T are transcription factors. And these are proteins which bind to specific sites on the DNA, and by that they control the ability to translate or transcribe certain parts of the DNA into genes. So transcription factors can act alone, usually they don't, or with other proteins, to either activate or suppress the recruitment of RNA polymerase to specific genes. And this is one way to present that. So we have the promoter downstream to the gene which is going to be transcribed. We have other components in the DNA like enhancer to which other transcription factor can bind and promote the transcription of a certain gene. Usually they work together with other transcription factors and either support or suppress gene expression. This is another way to present that, so we can get the proximal transcription factor binding site, and you can also get a more remote transcription factor binding site. Sometimes the transcription factor binding site can bind to a different chromosome, so they don't have to be just immediately next to the gene which is being transcribed. So how does it start? As always, we'll start at the beginning, and first of all, T cells come from the bone marrow. And we make him go through several steps to prove us that he wants to become a T cell, and he's dedicated to that purpose. So first, he has to leave the bone marrow and go to the thymus. There, it's going through several steps of maturation. Then it goes to the periphery and being activated into some effector cells. And this is another way to present that, a more complicated way. So basically, in the thymus, the T cell goes through several steps. Again, we want to make sure that it wants to become a T cell, that it has all the property that it requires to become a T cell. First, it starts with a double negative, which means it doesn't express CD8 or CD4. And during the first step, it has to arrange its TCR, the T cell receptor, first the beta chain, then the alpha chain. Then it becomes a double positive, both CD4 and CD8, has the full complete TCR, and then we want to test this TCR. These basically are two pictures which are showing the same. I just wanted to show you here that different transcription factors are expressed during different maturation stages. One of them is GATA3. So GATA3 is associated with Th2 response, allergies, and if someone wanted to knock out GATA3 and hopefully get rid of all the allergies, so yes, probably we won't get any allergies, but we won't get any T cells at all. So the phenotype will be a SCID phenotype. It won't be because GATA3 knockout is lethal, so we need GATA3. But at the same time, after the T cell has matured, we also want to select the T cells that we like the best. So first we start with positive selection, which means that we present the T cell different peptides that are expressed in the body and we ask them, do you recognize them? If they don't recognize them, then they just die of neglect. And if you think about it for a moment, we talk about a lot of autoimmunity and autoimmune diseases, but basically every T cell which comes from the thymus is autoreactive by definition. Because if it wasn't autoreactive, it wouldn't survive the first step of selection. So yes, there is a second step of selection, that's the negative selection, and we ask him, do you recognize it and do you know it too well? So if the TCR of a T cell create a very strong complex with a peptide, then it's been deleted, and this is the negative selection. So finally, we have a T cell which came out of the thymus. And then he has different options. We can ask him, what do you want to become now? Do you want to become a CD8 T cell that was actually decided in the thymus? Do you want to become some form of CD4 T cell? And you have different options, TH1, TH2, TH17. Tregs is another option, but you had to decide about it back in the thymus, so we're not going to talk about Tregs now. There's another form of Tregs, inducible Tregs, again, not this time. Just to show you what a CD8 is, and these are not CD4, these are several movies. The green one are the small rounded cells, are naive CD8, they were not activated. The big cells, the orange one, are CTLs, CD8s which have been activated. They acquired a different phenotype. They have all these orange granules which has the perforin and enzyme, all the things that they can use to kill the target. And if they don't recognize each other, which means none of them is directed against the other, then they just float around. They don't interact with each other. They can live perfectly well with each other. On the other hand, if they recognize each other, then you can see that there is no green cell left alone. Every green cell is being attacked by CTL. They form conjugates. And if we can take a closer look, what actually happens that when a CTL recognizes the target, first it forms a conjugate attached to it, then it polarizes all the granules to the contact area between the two, and then this green cell lost its color, which means that it died. The cell died, and after another several minutes, and that 1 hour conjugate, so that's a long time to form a conjugate, the second cell is dying as well. So this is indeed a killer cell, no regret, just moves on, if the body and goes on. So okay, our CD4 T cell wants to keep his hand clean. He doesn't want to become a CD8 T cell. He wants to become a T helper cell. He wants to help. And he has different ways to do that. He can become again TH1, TH2, or TH17. How can he become a specific helper cell? First, and I think yesterday I understood that this is the wrong slide because this is actually a CD8 T cell slide, but it's the same principle. So the cell meets an antigen presenting cell, it's being activated, then it proliferates and differentiates into a specific subset of T cell clone. And okay, so we put so much effort and time to educate our young CD4 cells, and now once it's out there, we want to understand what exactly it is. Are you a TH17, are you a TH1, or are you a TH2? How will we know what cell you became? And for that, we have the transcription factor. So we have the Rho gamma T in TH17 cells, Tbet in TH1 cells, and GATA3 in TH2 cells. And that's the reductionist approach, and thank you very much, we could have done with that. But So how the field had developed, or basically why do we need different T cells? So we need Th1 to eradicate intracellular pathogens and cause autoimmunity, I guess. We need Th2 to eradicate parasitic infection and to make a living. We need Th17 to eradicate extracellular bacteria and fungi and also to cause autoimmunity. And we need different regulatory cells to maintain self-tolerance and modulate the immune response to infection. And how did it start? So it started in the early 80s with two guys, Robert Kaufman and Tim Osman, and they asked the following question: Are B-cell help and delayed types and hypersensitivity reaction are mediated by different types of CD4 T helper cells? And they already called them T helper cells. So they ran different experiments. They had different cell lines and they tested different cell lines. First of all, that was the second step, but I think it make more sense to present it that way. They wanted to know if different cells can cause delay type hypersensitivity. And delay type hypersensitivity at that time is kind of a general term to describe any form of cell-mediated inflammation. So it could be the reaction to PPD, TB inflammation, multiple sclerosis, rheumatoid arthritis. Every process or inflammation which causes tissue damage will be defined as delay type hypersensitivity reaction. So they were not very nice. They took the cells and injected them into the food pads of mice, and they wait 24 hours and they measured the swelling of the food pad. And they saw that, and they actually divided the cell by that certain cell lines caused delay type hypersensitivity, tissue swelling, and certain cell lines did not cause similar swelling. So the one who caused the swelling were defined as Th1, and the one did not were defined as Th2. Then, actually that was a year before, they wanted to test the lines and see what kind of cytokines they secrete. So they took the cell, they stimulate them, and measured the cytokines in the supernatant. And there was a significant difference between the different lines. So the Th1 line secreted IL-2. I guess that the Th2 also secreted, but they just didn't see that. They all secreted IL-3, which is a general growth factor or support the survival and differentiation or survival of T cells. The Th1 secreted interferon gamma, which is the typical cytokine which they secrete. The Th2 cells secrete different things. They didn't know exactly what it was. They called it mast cell growth factor 2. It was called T cell growth factor 2. T cell growth factor 1 is IL-2. B cell factor, do you have any guess what it is today? So it's all the same cytokine. The mast cell growth factor, the T cell growth factor 2, and the B cell factor are all IL-4. So this is IL-4. And they also promoted Ig secretion and IgG1 secretion in mice. So they also supported B cell. And that perfectly answered the question. So you have one type of cells which promote delay type hypersensitivity, probably through interferon gamma secretion, and you have a second type of cells which secrete a lot of cytokines and promote B cells, and they don't cause tissue damage. Several years later, that was 1989, IL-10 was found, and it was found to inhibit Th1 function. So it was found that IL-10 was secreted by Th2 cell, and that was the general new concept at that time. So you get one cell subset which is able to support itself by secreting different cytokines, but at the same time it put a lot of effort to suppress the other cell type. So that was kind of a new concept. One cell is promoting itself and inhibiting the other cell type. And what are the effector functions of the different cells? So Th1 secretes interferon gamma. Interferon gamma activates B cell. It's kind of a simple way to present that, but activate B cell to secrete opsonizing IgG antibodies. Several pathogens. So pathogen can be engulfed and swallowed by macrophages. Interferon gamma also activates macrophages in two ways. It makes them express high MHC class 1, so they can say, hey, I'm infected, come and treat me. And at the same time, it can also push them to activate the lysosome and try to destroy anything that they already digested. It also activates CD8 T cell into CTLs, and then the CTLs can go to the macrophages or any other cell in the body which is infected with a pathogen and destroy it, as you saw before. The Th2 cells, they start with naive. Following some local stimulation, they start to secrete IL-4, IL-13, IL-5. They promote B cells to secrete neutralizing antibodies and also IgE. They promote and support the recruitment and development of mast cells and also eosinophils, all the Th2 response that we all well know, as we know. So the next thing that people ask themselves, okay, so we know what is the cytokine which is being secreted by the cells. It's interferon gamma in Th1 cell, and it is IL-4, mostly IL-4 in Th2 cell, but what makes them secrete this specific cytokine? This is early 90s. It's not that long time ago, so it's 20 years. It was discovered that Th1 cells are being supported by IL-12, so if IL-12 is provided, the naive cell will secrete interferon gamma. So interferon gamma is the readout. This is the results of Th1 stimulation. If you provide them with IL-12, they secrete interferon gamma. If you provide them with IL-4, they don't do that. Different combination of cytokines were provided. This is an interesting one. I think that if you give them both IL-12 and IL-4, you were able to suppress interferon gamma secretion, and people didn't have enough only with that, so they wanted to see if once the Th1 response was developed, or the substance was developed, whether the cell can maintain their phenotype, or whether the memory cells also have the same phenotype. So first they stimulated the cell, then they waited a week and stimulate them again, this time without the cytokines, just with CD3 to activate the T cell, and again, the same phenotype. If cells were stimulated first with IL-12, they will continue to secrete interferon gamma even when IL-12 is not there anymore. And I think that's an interesting fact. If they saw IL-4 in the process, somewhere in the activation process, if they saw IL-4, they won't become Th1 anymore. They won't secrete interferon gamma. So there could be some hierarchy there when IL-4 or Th2 response is superior to Th1, able to control Th1 response. Different combination of cytokine were provided, and this experiment described similar activation, but this time the cell were not activated just with cytokines, they were activated with APCs, and they also used APCs which were activated or stimulated with heat-killed Listeria monocytogenes, and they found that the HKLM-treated APCs also secreted IL-12 and were able to mediate the same Th1 expression or interferon gamma secretion. So IL-12 promotes CD1 differentiation, and the cell which probably provide IL-12 are the macrophages which are being activated by an intracellular pathogen. What drives Th2 differentiation? Basically IL-4. So if cells were activated in the presence of IL-2 and IL-4, and then re-challenge, only activated with beads without any cytokine, they would still secrete IL-4. So these cells were activated for different times, either 2 days, 3 days, or 5 days, and they were moved to another plate, and the cytokine production was tested, and cells which were stimulated with IL-4 actually secreted more IL-4. And this was also a new concept at the time, that IL-4 can promote IL-4 production. You have a cytokine, the cytokine is not only the product, but it's also the inducer. Interesting. And IL-4 was shown to play a dominant role in the differentiation. I already suggested that it might made a decision to which direction the cell will develop. So if cells were treated with IL-4, they would secrete IL-4. Interferon gamma production was shut down. If the cells were treated with anti-IL-4, then no IL-4 was produced, but interferon gamma was produced. So IL-4 plays a major role. It can either support Th2 differentiation, and when it's not there, the cell can progress and develop into Th1. 30
How
slides.
long ago was that?
This is, so that was 93, this is around 95, 96. So these are 30 slides already and not even one transcription factor. So this is the first one, and it's not any of the GATAs, Tibet or gamma T. This is 96, and this is a review by Abbas who described what was known at that time. So we start with the naive CD4 T cells. It meets the antigen which is presented upon an APC. It starts to proliferate, and then it can develop into Th1 or Th2, depends on which cytokine it was stimulated with. So if it meets IL-12, it will develop into Th1, and that was mediated. People already knew about cytokines, they knew about STATs. These are the transcription factor, signal transduction, and activator of transcription. So STAT4 is the one which responds to IL-12 stimulation. On the other side, if T cell will see the antigen in the context of IL-4, which can be secreted either by the T cell itself or by other cells, then through STAT6 it will develop into Th2. STAT4 and STAT6 knockout mice cannot generate either Th1 or Th2. So if we knock out STAT4, we won't develop any Th1. If we knock out STAT6, we won't develop any Th2. These are the first transcription factors. And a year later, finally, GATA3 came. So this is a very nice paper from Flavel's group in Yale, and he asked a very simple question. He asked himself, what is the difference between Th2 and Th1 cells? So we took the RNA of the two cells, they made DNA from them, and then they compared and checked what kind of RNA we have here that we don't have on the other cell. And they found that GATA3 was one of the genes which was transcribed in Th2 and not in Th1. GATA is a GATA binding protein, which means that it binds to a specific DNA sequence which has a GATA in it. It has a W on the other side or R on the other side. So W stands for either A or T and R for A or G. Basically, it has a GATA sequence in the middle, so it recognizes GATA. And they start playing with the GATA. So first they took a naive CD4 cell line and they incubate the cell lines under condition which promote Th1 cells. And they saw that naive cell already has GATA3. And that's an interesting point, I think. Naive cell already express GATA3 to begin with. And once they stimulate them to become into Th1 cells, then they lost GATA3 expression. The opposite happened with Th2 priming, so the cell maintained GATA3 expression, and accordingly the cytokine profile was changed. So with Th1 priming, no IL-4 production, although there was some IL-4 at the beginning. Difficult to see it here, maybe you can see it in the paper, in the pages. But interferon gamma expression was upregulated. With Th2 priming, no interferon gamma and IL-4 secretion. IL-4 is being transcribed. They next use the GATA3 antisense to knock down GATA3 within the cell. And these are two cell lines, both represent the wild type, and even numbers represent the transduced cell. And whenever they transduce the cell with GATA3, then the target cytokines were actually inhibited. So once cells were transduced with GATA3 antisense, IL-4 was shut down, IL-5 was reduced, IL-6 was also shut down, and the same for IL-10 and IL-13. And this is the northern blotting, so this is the RNA, and these are the protein levels. Transduction with GATA3 antisense basically abolish IL-4 expression, IL-6 expression, and significantly inhibits IL-5 expression. So GATA3 is not only important for 2H2 development, it's probably doing that, but by affecting the transcription of the different cytokines. Another way to show that, and that was during using an in vivo model, GATA3 was knocked out in a mice. Mice were vaccinated in different settings and re-challenge a week later. So that was again a way to test a memory response in an in vivo model to see if cells which developed into Th2 can actually preserve the phenotype in vivo and whether they can preserve it in memory cells as well. So you can see the cytokine profile in the inguinal lymph node. When mice were vaccinated, they produced interferon IL-4. They did not produce interferon gamma. And when GATA3 was knocked out, then there was no IL-4 expression. Instead, interferon gamma was expressed. So that was a shift from Th2 response to Th1. This is a general warning. This clinical data. So, but don't be too excited because this is in mice. Okay. So GATA3 is lethal. So without any GATA3, you can't get any mice or any product. And this is a conditional knockout of GATA3. It's not a conditional knockout. Actually, they made some change in the GATA3, so it cannot bind to the DNA at the same affinity. It basically binds to the DNA. Otherwise, we won't see any effect. And they use the classic asthma model in which mice are first sensitized to OVA and then challenged with OVA. They are being injected with OVA into the peritoneum and then get the OVA through inhalation. And you can see that in the non-transgene mice, there is inflammation in the tissue lung. You can see a lot of cells here. And once GATA3 is being knocked down, then there is less inflammation. They also did the bronchoalveolar lavage, and they actually measured the number of the cells, so less eosinophils in the bronchoalveolar lavage. IL-4 level, IL-5 levels, and IL-13 levels were also reduced in the transgenic mice. So GATA3 has something to do also in real life, in vivo. So this is the suggested model for Th2 activation. It starts with a naive T cell. It meets an antigen presented on an APC together with IL-4. IL-4 leads to STAT6 activation, which then goes into the nucleus. It induces the translation of GATA3. GATA3 then binds to other gene inside the DNA, and it activates IL-4, IL-13, and IL-5. So this is the STAT6 dependent pathway. But it's also there are also different other pathways which are STAT6 independent, which leads to GATA3 activation. So you can get activation through STAT5A phosphorylation, and this can be induced by IL-2, but also through IL-7, and I think the new guy in the neighborhood is the TSLP, which can also induce Th2 differentiation through STAT5. Once STAT5 is being phosphorylated, it can induce IL-4, and once IL-4 is being prescribed, then we move to the other side of this slide. IL-4 is being produced. Positive feedback, autocrine pathway, IL-4 simulation, STAT6, and again downstream activation of Th2 response. Another thing about an alternative way to induce Th2 subset is through the TCR activation. So TCR activation by itself can induce through NF-kappa B. I think we can't give any talk about transcription factor without mentioning NF-kappa B. So it can induce NF-kappa B activation, which again would lead to the same pathway. But there is something special about this TCR activation. It was shown that TCR would lead to Th2 differentiation when it meets peptide with low affinity, which means that only with a weak stimulation, cells will develop into Th2. With more strong stimulation, they will develop into Th1 cell. And it makes you wonder why we see more allergies, and I'll talk about it at the end. That's a short break. Th1. So we know what we know about Th1 by now. This is already 2000. So we move forward. We know that the Th1 cell are stimulated by interferon gamma and IL-12. They secrete interferon gamma as the end product or the cytokine. And the question is, if Th2 cells have their own special transcription factor, what transcription factor Th1 cells are using? So that was the second group. What they first did was to read Flavel's paper on GATA3. And they asked a different question. They didn't ask what is the difference between Th2 and Th1. They asked what was the difference between Th1 and Th2. So they did the same thing. They checked the RNA, and they wanted to ask what we have in the Th1 that we don't have in the Th2. I'm sure that Flavel is very upset about the fact that we wasn't doing the same at the same time when he first identified GAT3. But this group of Glimker identified TBET as the master regulator of Th1. TBET stands for T-box expressed in T cells. T-box is a group of transcription factors which were involved, known to be involved in limb and heart development. And they, yeah.
So what kind of assay were these in the previous paper doing to see what was different in terms of transcription?
They were doing representational difference analysis. I was practicing on it, so let me see if we have it here. But it's going to take a minute. So you have your DNA, one cell, I guess. It's quite better at home, sorry. And you have a different DNA of the other cell. This is the DNA. And different cells are making different RNAs because it depends on the cell. So cell A will make these RNAs, and cell B will make these RNAs. And you need to check cells which are close to each other, otherwise you'll get too much of a difference between the different genes which are being expressed. So you first isolate the RNA and you translate it into cDNA, and then you ask, what do I have in one cell? So you mark the RNA of the cell that you are interested in with a different marker, just tag them another DNA sequence that you can recognize, and then you mix them together. But you mix them when you have a lot of access, significant access of one over the other. So you can take, let's say, 1 microgram of this RNA product and 100 microgram of this RNA product, and then you mix them together. So every time that you have the same RNA, during the first step of the PCR, they will attach to each other. And then you start your PCR reaction with a primer which fits the tag that you added. So that will be here, and it will be here, it will be here. But once you start the PCR reaction, then you will get no product here because it was already blocked by the other RNA. So this one, and this one would be blocked, and the only product that you're going to get is this one. And then you sequence that, and you go to the IGV website, and you type your sequence, and you get the product what gene it is in.
You're looking at the whole genome, but basically only things that are either all or nothing in terms of expression. You can't see small.
These are not whole genome, these are RNA, so it was already described.
Right, a whole transcriptome.
Yes, exactly.
You're looking at whole transcriptomes, but you're only looking at things that are all or nothing in one cell type or the other.
Yes,
You can't see.
and you can do it at different levels. So you can run it once, and you can run it several times, and depend on the level of transcription. You'll need to run it different times or for longer periods to see a difference. But GATA3 and TBET were observed during the first cycle of this assay. This is the cool assay. It's very nice.
Yeah, no, it predates
Yeah.
a lot of technology out there now.
I asked you to ask the question, so.
That's not true, actually. I'm glad you were prepared for it.
So TBET was recognized, and they couldn't do exactly what Flavel did, so they did something a bit different. They stimulate the cells under Th1 or Th2 conditions, either with IL-12 interferon gamma or with IL-4, and they check for TBET expression. So only with under Th1 stimulation condition, then TBET was expressed. They then formed retroviral vectors to induce forced expression of TBET. It was marked with the GFP, some green fluorescent protein, so they can see that the cells were actually transduced. One of them had the TBET in them, and one of them did not have the TBET in them. And then with forced expression, they wanted to see what is the result. So if they took the cells and stimulated them with interferon gamma, when the cells were forced to express TBET, they also expressed higher levels of interferon gamma. So this is a positive feedback with interferon gamma stimulation. If cells were stimulated with IL-4, these are Th2 condition, and they were forced to express Tbet, then IL-4 production, which should be the natural response, was blocked. So instead of producing a lot of IL-4, IL-4 was suppressed. And I saw the presentation to my wife last night, and she told me that it looks like Rorschach staining, and she doesn't exactly understand what they mean, but fine. And then they challenged it further on. So they didn't use a naive T cell. They took Th2 lines, the one that were already recognized 15 years before, and they wanted to see what happens to Th2 cell lines when Tbet is forced to be expressed. So again, IL-4 was shut down. IL-5 was also shut down. This is starting from 77% of the cell producing IL-4 down to 26. And they wanted to make sure that it's not secondary to interferon gamma secretion, so they took the same Th2 cell. This time, this cell didn't have the interferon gamma receptor. And again, with forced Tbet expression, IL-4 was shut down and IL-5 was shut down, which means that Tbet is able to shut down IL-4 and IL-5 production at the nucleus level. So it has something to do with the transcription of IL-4, IL-5, and probably other Th2 and cytokines.
Does that mean you can change the phenotype of a differentiated T cell after it's decided what it wants to be?
I think the answer today is yes, and that's kind of that's the latest news in the field, I guess. Basically today we know that you can change any cell. You can start with a skin cell and you put on them 4 transcription factor, and you can induce them to go back into iPS, pluripotent stem cells. And you probably can do the same. And there are new evidence to suggest that once the cell was already pushed to develop into a certain subset, and that's why I quote when I say differentiated, because they are not really differentiated. Probably once they become memory cells, it's easier to make them change their subset with the different cytokines. So if you had one single cell develop into a Th2 cell, and then you stimulate it again with a different cytokine, you can push it to develop into a Th1 cell. And I think that probably one of the things that you're doing with immunotherapy, if you have an allergen which has low affinity to TCR and develops into Th2 cell, what we do with immunotherapy might do is to push them into Th1, because you produce at a higher level, higher concentration, I guess, of the peptide than it's usually seen in the environment, even though we do it to start with a very low dose. That could be one of the things that you do with immunotherapy. But yes, the answer is yes. With different cytokines, you can push already differentiated cell to produce different cytokines.
Well, we've interrupted you. Maybe it's a naive question. We talked about GATA3, which implies to me there must be a GATA1 and 2.
And 3 and 4 and 5 and 6.
They all do different things?
There are 6 GATA. The first 3 ones has different roles in the hematopoietic stem cells, and 4 to 6 are in other tissues. You mentioned GATA1, which has to do something with red blood cells, but also with mast cell differentiation. And another interesting thing about GATA3, if you use force expression of GATA3 in the early double negative phase of the T cell, you can make them acquire mast cell phenotype. Even though they are coming from a different cell line, the mast cell progenitor, you can actually push double negative T cell to produce different, to acquire the shape of a mast cell and even produce mast cell products. So it depends on when the cells express the transcription factor and at what level.
Do you know, have they been able to crystallize GATA3? We had Sanjeev Jain was here in our section. That was his project trying to do that.
I don't know. Yeah, I mean the structure. You can check the structure. I think they did, because I saw the structure of the GATA3, so it was well described. I didn't specifically look for that. And they have 2 zinc fingers attached to the GATA, and so there's a lot of discussion about the structure of the GATA genes in general.
Because I guess the goal was to find an active site, or so you could have soluble inhibitors of it to skew the immune response.
I don't know exactly what was the goal. I think it's really interesting to know where it binds to. I think we have new methods to know today where it binds to, and we can use the paint again. But definitely people were looking for targets. Yeah.
Another naive question, but with today's technology, do they ever go back and sort of repeat these experiments and try and find out whether the factors that they think are important are the right ones?
They are doing that, especially with epigenetic change. So I have a few slides which I wasn't planning to show, but I can show you that later. I think another paint I was also practicing. So another way that you can do it today, so you can take the DNA again. DNA. You can break it with restriction enzyme, and then you can get different DNA parts. Then you assume that different transcription factors are already bound to the DNA when you use it. So different DNA areas. I'm sorry, that's not quite Russian. Different areas will have different transcription factors. This will be, let's say, GATA3, this will be TBET, and this will be another transcription factor. And then you can come again with an antibody, and you have a specific antibody. This time you can just use the antibody again, one transcription factor. So if you have GATA3 and you want to know where on the DNA GATA3 binds to, then you can take your antibody. And then, basically similar to ELISA, you can take only the DNA which was bind to the antibody, and you sequence that part of the DNA, and then you can go and compare it to the human genome and see where exactly it was binding to. So now it's called chromatin immunoprecipitation. You can take the chromatin, bind it to a specific use antibodies against specific transcription factor in different cells, and see where the transcription factor were bound to. And people are doing that, especially when they study different epigenetic changes, to see if in different cells, different areas of the DNA are open for transcription, and see which transcription factor are bound to each part of the DNA. So yes, it was repeated. Where was I? I think we can skip that one. Again, clinical studies, I'm sorry. No, actually I wanted to show that because this is the same group of Glimker from the first one we described, TBET, and I think the first paper was in Cell 2000, and then a couple of years later, this is 2002, they came up with a Science paper. They knocked out TBET, and they saw that once TBET was knocked out, CD4 response could not be suppressed. So if they took CD4 T cells, naive T cells, they stimulate them with different cytokines. So the wild type, this was stimulation with Th1 condition. So in the wild type, you got interferon gamma secretion. In the knockout, there was no interferon gamma secretion, but also in the wild type, IL-4 and IL-5 were suppressed, and in the knockout, they were not suppressed, so the cells expressed Th2 cytokines, and this is when they used Th1 cells, cell line, and again, they saw the same thing. Once TBET was knocked out, then instead of producing interferon gamma, and these are already Th1 cells, this is something to do with the reprogramming that you were asking about, then they were secreting IL-4 and IL-5. This is a Science 2002, the same group, and they had the nerve to publish a back-to-back page paper in the same Science journal, another paper which actually checked the effect of TBET in vivo. So first they went to human patients and they checked biopsies from healthy patients. I guess they were not healthy because they still had underwent bronchoscopy, and asthma patient, and they checked to see if there was CD4 infiltration in the biopsy. So yes, basically the same number of CD4 cells, but when they stained the cells for TBET, so TBET were expressed in normal controls, but in asthma patients there was no TBET expression. So this is healthy control 1, healthy control 2, significant number of TBET positive CD4 cells, but in the asthma patient, no TBET cells, positive cells were found. And when they generated a mouse model with TBET knockout, they saw that at the age of 6 to 7 weeks, the mice developed spontaneous asthma or hyperreactive airway disease even without overstimulation. The natural history was development of asthma phenotype. So these are healthy lungs. These are the heterozygous mouse, and these are samples from a Tibet knockout mouse. You can see the infiltration, you can see the hyperplasia in the airway and the inflammation in the airway. So Tibet definitely has something to do with suppressing Th2 response, and when it's not there, then the natural result will be Th2 response. So Th1 are the master Tibet are the master regulators of Th1 response. Interferon is the main cytokine. IL-12 promotes Th1 response, and Tibet is the master regulator. A different way to show that, we start with a naive T cell. It meets the antigen on an MHC. I talked about IL-12, but you can see here that during the initial activation phase, IL-12 doesn't play a role because the IL-12 pathway is being shut down. And in my idea, it could be a way to try and prevent from the development of a natural or spontaneous Th1 response, because the Th1 response is much more aggressive. It can cause tissue damage. So maybe we want to suppress it until it's really needed. But once the cell were activated and Tibet is already expressed, IL-12 receptor is being expressed, interferon gamma is being secreted, and there is a positive feedback loop, and also ready to respond to more IL-12 which will be secreted. But Tibet is it's not the only thing that it's doing. It's not only secreting its own cytokines, it also put a lot of effort to suppress the development into other Th subsets. So it's not playing a fair game. It's not only secreting its own cytokine, at the same time it's also suppressed GATA3 transcription, it's also suppressed Rho gamma T, but we're not there yet because nobody knew about Rho gamma T at that time. So we have a Th1 and we have a Th2. We can explain tissue damage and we can explain antibody production. Why do we need Th17? And the reason is because we were not able to explain everything just by the Th1 and Th2 hypotheses. So the question that was asked, are B cell help and delayed hypersensitivity reaction are mediated by different CD4 T cells. And delayed type hypersensitivity, again, it equals tissue damage. So any inflammation which causes tissue damage will be defined as delayed type hypersensitivity. Most of the autoimmune processes will be defined as delayed type hypersensitivity. EAE, which is an animal model for MS, was also defined as DTH. And since interferon gamma is the central effector Th1 cytokine, we would expect that if we target interferon gamma, then we'll be able either to prevent, or if we give interferon gamma, we'll be able to promote EAE or MS. But that wasn't actually the case. So when tested in animal models, so mice which were treated with antibodies against interferon gamma actually had a more severe disease instead of preventing the disease. When rats were treated with interferon gamma instead of causing the disease was suppressed. Same thing was observed with the model of rheumatoid arthritis. Treatment with gamma interferon actually prevent arthritis from the development of arthritis instead of promoting the disease. TNF, which is another Th1 cytokine, also had a potent anti-inflammatory effect in MS model, animal MS model. And this is already 1998, so it's pretty late. This is a very nice review by Steinman. If you have the time, I would suggest that you look it up and read it because it's very nicely written. I'm sure that he lost a lot of friends after he wrote what he wrote, and basically was blaming all the Immunology Society for ignoring all the evidence that were out there. They're so fixed to the theory of Th1 and Th2 that we didn't even stop to think whether there is another cell type in there. Watch. Just to be honest, so in humans, it's not exactly the same. If you treat MS patient with gamma interferon, yes, you exacerbate the disease. If you treat MS patient with anti-TNF, then as we all know, you can cause exacerbation of the disease, and anti-TNFs are contraindicated in MS. I didn't know about it. And when people actually later, it was 2003, wanted to check whether there is a new subset of T cells which promote auto-inflammatory or inflammatory processes, then they finally came to the conclusion that there is another TH subset. So the first thing, when you need a new TH subset, you want to ask yourself what cytokine promote the activation of this subset, and it was found to be IL-23. And that was part of the confusion, I think part of the confusion between Th1 response and what later known to be Th17 response. Because IL-12 and IL-23 share one subunit, it's called P40. And where first people tried to see if Th1 is actually responsible for a lot of the inflammation or the inflammation in autoimmunity models, then they knocked out P40. If they knocked out P40, then yes, they knocked out IL-23, but they also knocked out IL-12. Or the other way around, they knocked out IL-12, but they didn't know that they were also knocking out IL-23. So this is brain sections of an EAE model, and you can see that when you knock out P40, you don't have any IL-12, you don't have any IL-23, then the brain tissue is clear. There is no inflammation in there. If you knock down only P35, only the IL-12, then you still get the same inflammation. If you knock out P19, which is specific only for IL-23, then you can prevent the inflammation. This is an heterozygous knockout for for P19, so kind of an intermediate response. So IL-23 is the responsible for the promoting cytokine for the new TH subset. And that was the same with rheumatoid arthritis model. When you knock out P19, then no arthritis was developed. If you knock out only P35, you can actually get more severe disease, maybe because you turn the direction more into IL-23. Then comes the induced cytokine. People ask themselves, okay, we know that IL-23 produce something or support the production of some cytokine. What it is, people knew at that time about IL-17, and it was found that IL-23 promote IL-17 secretion. Later on, it was found that TGF-beta and IL-6 can do it even better. So a new subset of CD4 T cells was announced, basically the same by two groups at the same time. Dr. Park is here at the Immunology department. I think she's still here. And this is another group which basically describes the same TH subset. What they first show that if you take TH1 cell lines or TH2 cell line and you stimulate them with IL-23, you can't induce IL-17 expression. So these are not TH1 and they are not TH2. These are a new kind of cells. In order to induce IL-17 expression, yes, you need IL-23, but you also need to suppress TH1 response. So you need to treat the cells with anti-interferon gamma so they will express IL-17. You can do it either by supplying the cells with anti-interferon gamma, but you can also knock down STAT1, which is downstream to interferon gamma, or you can knock down TBET. This is the gene of the TBET. So TH1 blocked IL-17 secretion. And now everything is okay. It makes sense because if you, yes, it's very simple. Because if you promote TH1 response, you can get more interferon gamma. Interferon gamma actually suppress TH17. So whenever people were treating animals with interferon gamma, or they were actually promoting TH17, and they made the disease worse. And if they blocked interferon gamma, they blocked interferon gamma, then there was no TH17 suppression, more severe disease. Again, interferon gamma suppressed the disease, anti-interferon gamma promoted the disease, and now everything makes sense. But IL-4 can do the same. So if you stimulate the cells with IL-4, again, you block IL-17 production. And it seems that if IL-17 is so important and so I don't know if it's so important, but so dangerous to the animal, then you need to suppress it. So you can either suppress it very easily by promoting the response toward TH1 or TH2. Then we need a master regulator, and or gamma T was identified as the regulator. They didn't do any sequencing. They already knew about IL-17. They knew that there are a lot of IL-17 cells in the gut. They already knew about oral gamma T, which was expressed in different tissues. So they just made the connection between oral gamma T and IL-17, and they showed that saw that once you knock out oral gamma T, even with stimulation which should support IL-17 production, IL-17 production was shut down. Another intracellular signaling pathway. So it starts with IL-6 or IL-23 or IL-21 later, which first lead to STAT3 phosphorylation. This leads to oral gamma T transcription, and again, self-activation. IL-17 is produced, IL-21 is produced, IL-22 and TGF-beta, which again activate the cell and positive feedback. So now that we have Th17, why do we need them? So Th17 cells are located in the lamina propria and different mucosal sites and the skin. They secrete a lot of cytokines which promote granulopoiesis. Th17 receptor is widely expressed, and once it's been activated on different cells, it induces secretion of TNF and many inflammatory cytokines, which also attract granulocyte to the site of the inflammation. So you get the invasive pathogen, and you recruit all the cell into the site, including granulocyte and neutrophils. IL-17 can also induce the formation of secondary lymphoid tissue. So it creates like secondary lymph nodes in the site of the inflammation, which recruit more cells, also activate T cells, and you have your antipathogen T cell, macrophages, neutrophils, and you also have the B cells and the antibodies all in the same site, so it creates its own base right next to the site of the inflammation. They can augment Th1 response, and they protect us against fungal infection, especially mucocutaneous candidiasis, by stimulation of pattern recognition elements on macrophages and disease. That's why when you don't have Th17 cells, the result is CMC, chronic mucocutaneous candidiasis, and IL-21 also promotes B cells. Why we don't need Th17 cells? We're almost done. We're not almost there. So Th17 cells can induce a lot of tissue damage, either by direct TCR recognition, I guess it's fast ligand interaction, but they can recruit a lot of myeloid cells to the site. Th17 cells were associated with MS, psoriasis, Crohn's disease, rheumatoid arthritis, uveitis, and also asthma. So whenever you have an inflammation, Th17 cells are there. And they also might have a role in cancer, either by inducing chronic inflammation or directly stimulating metastasis and tumor progression. So how we think it works? It starts with an antigen recognized by a TCR, which leads to the classic transcription factor. But we also have the cytokines, different cytokines around us, which trigger STAT phosphorylation and translation of cytokine receptors and cytokines, and also the master transcription factors. They in turn cause a positive feedback loop to produce more cytokines and cytokine receptors, and so on and so on. Another way to show that we have different cell lines with the Th1 starts with interferon gamma stimulation, STAT1 phosphorylation, Tbet. The next step will be IL-12 secretion, IL-12 receptor, and STAT4 phosphorylation. So this is the combination of cytokines, STATs, and master regulators in Th1 cells. And people are trying to say now that there are different stages of maturation. You have the first step of activation, when the first set of transcription factors are being expressed, and then you get the second phase, when the cell needs to be committed, needs to get committed to become either Th1, either Th2, or Th17. So these are the different combinations of cytokines. And now we'll spend the last time, another 5 minutes to go over that, but no, so that's basically describe the interaction between different Th cells, but there is a simple way to present that. And I guess that if we start with Th2 cells, then yes, the first step will be STAT6 phosphorylation, GATA3 expression, STAT5 phosphorylation, and yes, the cell is promoting Th2 differentiation. But at the same time, it put a lot of effort into suppressing other Th cells formation. So GATA3 will suppress STAT4 and will suppress Tbet expression. Th1 will try to do the same. It tried to suppress GATA3 expression. So it blocked the differentiation of Th2 cells. Th17 are actually being suppressed by Th1 and Th2, and they don't suppress other cells. They have some interaction with inducible Tregs, but basically they're not known to suppress Th1 and Th2. And again, it could be because they have a very strong effector and they can induce strong tissue damage, and maybe you want to prevent that. But I think what makes more sense is just we didn't recognize the pathways which actually do the same to the other cells. Do you have another 5 minutes? Or?
Well, I mean, people have to leave, will leave. Otherwise, just go ahead.
So I guess no gene or cell type is important enough unless you have a disease associated with this specific cell type. So are there any abnormalities known in Th1 differentiation of function? So sadly enough, there are no known defects that block Th1 differentiation yet. I'm sure there will come. But there are different defects in interferon gamma signaling pathway and in the IL-12, IL-23 pathway. And they lead to Mendelian susceptibility to mycobacterial diseases. So defects in interferon gamma receptor, IL-12 receptor, STAT1 will lead to susceptibility to mycobacterial infection. Basically, T cells either cannot secrete the interferon gamma, or the macrophages which are supposed to respond to the interferon gamma cannot respond to interferon gamma. The other way that there is a positive feedback loop when the activated macrophages can secrete IL-12 and the T cell cannot respond to the IL-12. So whenever we have problem in this pathway, the result will be susceptibility to mycobacterial diseases. Another type of diseases are the STAT1 diseases, which Matt talked about several weeks ago. And we know that complete deficiency STAT1 will prevent Th1 response and susceptibility to mycobacterial infection, as I said. But there is also the gain of function mutation, and I finally understand how it causes the same effect. So when you have gain of function mutation, we have constant Th1 activation, we have constant interferon gamma secretion, and interferon gamma secretion suppress Th17. So constant Th1, we promote Th1, we inhibit Th17. And if we don't have any Th17, then we don't have any antifungal Th cells. And the result is chronic mucocutaneous candidiasis. And that's why when you treat this patient with GM-CSF, you can bypass the defect because you can replace some of the cytokines which are supposed to be replaced by Th17 cells. And it's probably not true because there are other explanations for that, including IL-27 secretion. But for me, it makes sense now to explain that by interferon gamma secretion, inhibition of Th17, and no cells to fight fungal infection. Th17 defect, I think it's the most known defect. It's STAT3, mutation in STAT3 causes hyper-IgE syndrome, and it was later shown by Josh Miller that these cells don't have any Th17, these patients don't have Th17 cells. So if you check for Th17 expression, you can see that patients with hypereosinophilic syndrome don't express IL-17 positive cells. What about Th2? And I think as allergists, we should feel ashamed that we don't have any diseases associated with Th2 differentiation. So we know that allelic mutation in GATA3 are lethal, so we can get human without GATA3. There is haploinsufficiency in GATA3, and as we all know, haploinsufficiency causes HDR. This is hypoparathyroidism, deafness, and renal dysplasia. And no, any of these patients, of the 5 patients which were described, or the 3 families, they don't have any major immunological abnormalities. So we can't blame GATA3. But as you remember, I talked about the fact that weak TCR signaling favors Th2 response. And some patients with mutation in the associated PCR signaling show characteristic of skewed Th2 response. Patients with mutation in ZAP70, for example, they can present with eczema, elevated number of Th2 cells, elevated IL-4, IL-5, and eosinophilia. Omen-like syndrome, basically an Omen-like syndrome. And I heard that there is a new patient now in children with similar presentation. So if we see patients with more severe eczema, which is difficult to treat, do we need to check them for nitrogen stimulation? I guess the answer is yes. We at least need to think about it and look for it. And should we sequence them? I think we always need to sequence something. But the question is, what do we need to sequence? So we can go back to this pathway. If there is a STAT1 gain of function mutation, why wouldn't be a STAT6 gain of function mutation, which will lead to only GATA3 expression and high cytokine profile? What about gain of function in STAT5A? What about mutation in TBET, which prevent GATA3 from being produced? Because TBET can suppress GATA3 at the DNA level, but it can also just bind to GATA3 and prevent it from going into the nucleus. So maybe there is a problem in the site where TBET binds to GATA3. So there are different genes that can explain severe forms of allergies, but I think that we just need to look for them. Just several comments. When I was preparing this talk, I was thinking about the fact that maybe Th2 differentiation is the default. IL-4 is expressed in naive T cell, and IL-4 produce more IL-4. So it's push the cell to develop into Th2 T cells. And even when cells are stimulated by IL-12, when they are stimulated also with IL-4, then it blocks the differentiation into Th1. GATA3, as Flavel showed, was also expressed in naive T cells, so it's already there to begin with. And GATA3 is activated upon weak TCR signaling, and I assume that most of the TCR signaling or TCR interaction with peptide are weak, because we don't want the immune system to be activated all the time. So maybe that is the default, and maybe that can also explain the Hagin hypothesis. When you don't have a lot of strong TCR activation, the default is Th2, and that's why we see more and more allergies. So to summarize, the distinctive differentiation states of various CD4 effector subpopulation are determined largely by the set of transcription factors they express. The induction of different genes can be achieved in several ways, and at least in vivo can stimulate or decide which gene will be expressed based on the cytokines the cells are seeing while they are being activated. And final request, please don't blame it on the CD4 cells. CD4 cells are only responders, so they will respond to whatever cytokine they will see. And that raises another question, what should we target? Because in the last several weeks we heard a few talks about different biological treatment to target different cytokines. I was mentioning that maybe immunotherapy is doing something similar, just change the cytokine environment. But maybe we are targeting the wrong thing. Maybe instead of trying to target the process here, once the T cells were already activated, maybe we should target it here before when everything is starting. So again, TSLP would be a perfect candidate for that, and maybe other cytokine which start the first step, differentiation, should be the target. You can read it yourself actually. So this is from the same review by Steinman. He was really upset about the fact that it took so many years to discover that there is another Th17 subset. So immunologists have displayed a long affection for sociobiology and population biology. They talk about T cell, the young T cell, the way they develop, the process they have to go through. So not only that we define various population of subset of T cell, we assign them personalities. So we ascribe qualities to T cell, like altruism, we call them helper cells. We used to call them suppressor cells, and now we give them the authority and we call them regulatory cells, and we also call them killer cells. So they are there, they're just responding to different stimulation, and that's it. That will be for the next time. I think that I didn't talk about natural Tregs, usable Tregs. There is a new subset of Th cell, T helper cells. They are called T follicular helper cells. They have a different transcription factor. I didn't talk about Th9 and Th2 basically because they are not in Wikipedia, so I guess they don't exist. There are a lot of epigenetic changes. If somebody wants to stay, I can also show you about something about the epigenetic changes. And also plasticity. So once they have been committed to certain subset, they can still go back, especially when they turn into memory cells, they can switch back to a different subset. So thank you very much.
So way back when I was studying these cells, we used sheep red cells to separate out the T lymphocytes. What were they binding to on the sheep red cells that made them,
I think back then, I think Rebecca Buckley at Duke is still doing that. She separated the cells. At least 5 years ago, she was still doing that. So you separate the cells through different processes. You start with peanut agglutening and then soybean agglutening. There are different lectins that are on the cell surface, and I guess that there are just different lectins on the T cells, which are attracted to the red chips. And actually, yes, they attract.
Have sheep red blood. They call them rosettes.
Yes, exactly.
The
Some
size of
rosette.
little sheep.
So the first time when they first wanted to pure bone marrow, they started with the whole bone marrow. The first step was peanut lectins and then soy lectins. And I think I told you several weeks ago that it's kind of strange that you have a lot of peanut allergies because peanut actually directly can activate T cells. So maybe that's the reason. Prepare them in different ways, and maybe that's caused T cell activation. The last step of bone marrow purification to make sure that there are no T cell left, that was the T cell, the red blood cell, the sheep red blood cell rosette. They attach. Sorry, yeah.
Are those more pattern recognition receptors then, or
On the
T cell?
I think these are lectins. There are sugar moieties which attach to the red blood cell, but I'm not sure what it's.
David, other than the fact that a GATA3 biallelic mutation is lethal, is there any other diseases where there are no Th2 cells present?
There is there is a spontaneous disease when you don't have any GATA3. THT cells just disappear. I don't know what the genetic cause
Do
for
they
that.
have any disease entity when they all disappear?
They have more autoimmunity.
So that's the risk if you get rid of Th2 cells altogether, then autoimmunity is.
But there is
Yeah, just sort of to make a pitch for the sort of research I'm interested in. I mean, the last quote you give there, I think is a good point of how, and it's not just immunologists, but all science, we hang on to the bias. We study what we know already is true. And a lot of the experiments that build these pathways are based on what we already know and are sort of maybe not relevant to what's actually happening in the system because they're ex vivo, they're out of they're ex vivo and they're sort of using non-physiologic properties. I'm curious because there's a very similar story with hematopoiesis where we worked out all the transcription factors and so forth, and then people have gone back and used a system like a whole genome expression array and stuff of different cell populations and found a whole new, like double, triple the number of transcription factors that seem to be involved and things like that. Has anybody used that sort of approach, just taking different populations of T cells and other immune cells, looked at whole, RNA sequencing or transcriptome microarray profiles?
I think what people are doing today is a chromatin immunoprecipitation. So this is the chromatin, it's being packed, the DNA is being packed in different forms. The basic form is the nucleosome, which are 156 base pair, around 8 histones, and there are different acetylation or methylation side chain which can be bind to the histones, and based on that you can affect the DNA and decide whether it will be open for transcription or not. And people test the different TH1, TH2, and TH17 locus. So that will be, for example, the TH. This is the transcription sites of IL-4, IL-13, and IL-4, upstream and downstream to the genes themselves. And you can see the arrows mean that this is a DNase hypersensitive site, which means if you take the DNA and you add DNase, it will cut the DNA there, and it basically means that this area is open for transcription. And you can see that in naive cells, you don't have a lot of DNase sites in this region, but once you check that in TH2 cells, then you get a lot of sites like that. And if you check the nucleosome itself, you can see that green suggests that the histone is open for transcription. So there are a lot of regions which are stained with green in the TH2 cell, not a lot of region in the naive cell, and actually turned blue, which means no transcription in the TH1 cell. So these are the epigenetic changes. You start with the transcription of GATA3, for example, and GATA3 is first expressed, and then it goes to its own site, and it open the site for transcription. It goes also to the Tibet site and it closes it for transcription. So it recruits other enzymes which are responsible for histone methylation or acetylation, and it said, okay, I'm becoming a Th2 cell now. I want to promote my own cytokines, but I also want to block all the other cytokines. So you can see the same within each and every cell type. So this is the interferon gamma site. Th1 cells, it's open for transcription. In Naïve cell, it's not that open, and it's totally blocked in Th2 cells. So Tibet is doing the same. It's open its own cytokine, but it blocks other cytokines. And this is for IL-17, for Th17 cells. So I told you that it's not regulating other cells, and you can actually see that because also in Th2 and Th1 cells, there isn't a significant change. It wasn't blocked. So it probably has less effect on other cell lines. And although there are these changes, so once the T cell is becoming a memory cell with recall response, the second time it meets its antigen, you can use different cytokines. And again, instead of staying a GATA3 cell, Th2 cell, it can become a Th17 cell or Th1 cell, and it still express the two transcription factors. So it can express both GATA3 and Rho gamma T. So that's why the master regulator genes are not really master regulators. It was also shown for Tregs. So Tregs can express both FOXP3 and also Tibet, Rho gamma T, and GATA3. And I think Dan Campbell here showed that in order for Tregs to suppress Th1, they need to express the Tibet. So maybe it also caused them to express different chemokine receptors, so they get to the same place in the body, but maybe it would be another way to direct the T cell to where they need to go. So if I have inflammation in the gut, I need a Th1 response in the gut, I will express Tibet and my T cell will go there. The FOXP3 cell also will be induced to express the same Tibet transcription factor. They will also express the same chemokine receptors or integrins, and they will go to the same site to control the inflammation. So on one hand, the inflammation is being induced. On the second hand, it's being controlled. So there is no master gene.
I know this is maybe not what you directly asked, but if you look at a fairly local immune dysregulation like a nasal polyp, does anybody look to see whether this is a cytokine dysregulation or is it a cellular
I
dysregulation?
didn't look into that. I don't know.
Yeah, when you were reading this, do you have any idea in the evolution at what point it became this complex, and when evolutionary just had a T cell and not all these subsets, or how this developed?
I think somewhere in 1997 when Flavel actually started.
Well, actually, where the adaptive immune system evolved was cartilaginous fish.
I read somewhere, I think it was 5, I don't remember exactly the species in which it was developed.
The first adaptive immune responses are after cartilaginous fish.
Okay.
T cells evolved.
How many million years ago was that?
Probably before 1997.
That's when we lost
What
control.
was the word of the day? Somebody texted into me.
Transcription.
Transcription?
Yes.
Well, next year I'm going to give you a harder topic.
I just wonder if anybody
You don't
that's
have to
other
listen,
than
that's
Bill
all.
understood what he just said.
Nick, who's been retired like 10 years, turned to me and said, "This is the first time I've ever understood this stuff." So you did a great job.
Yes.